Alright, welcome back to this channel. Let us talk about genetics. Now in genetics, we are basically talking about genes. We are basically studying genes. Is that okay? Now we said genes are basic unit of what? Hereditary. Is that okay? And what is hereditary? What is heredity rather? Now there are two terms that we are likely to confuse. Heredity and hereditary. Is that okay? This word here is a noun, and this word here is an adjective. Is that okay? So a noun, heredity is simply the transfer of inheritance from parent to offspring. We are going to get there. Just calm down. I'm going to give you notes. Then hereditary means something that is inherited, that can be inherited. And what can be inherited? We call them traits. Is that okay? Traits or characteristics. Is that okay? But in order to understand how these genes are passed on from parent to offspring, we have to first understand the concept of meiosis. Meiosis. Are we good? So briefly, we are going to go over meiosis in 1 second. Are you with me? Now, meiosis, just like the other cell division, which is mitosis, it begins with what? With prophase. Meiosis begins with what? Prophase. Now, in the prophase, there are other stages in prophase. We have do you remember? Let Zion play. Have I not talked to this before? Leptotin, zygote stage, pachytene, and diakinesis. Alright, these stages, is that okay? These are the stages in prophase. That's why prophase is the longest stage in meiosis. Is that okay? What we are getting at is how inheritance is transferred, how traits are transferred from parent to offspring. That is inheritance, how inheritance occurs. Alright, so in the leptotin stage, we mentioned that the things that happen are the chromosome thicken. So you must know what the chromosome is. We are going to explain when we start the concept of genetics. The chromosome will thicken and condense, and replication first takes place ahead of that. So after replication now takes place, it now begins to thicken. Then as it thickens, the chromosome will now assemble themselves in. First, they were in dyads, then they now assemble themselves in tetrads. They assemble themselves in what? Tetrad mean, you see these 4 legs here? That's a tetrad. Is that okay? This is a dyad. This is a dyad. Is that okay? Dyad simply dyad is what we have in mitosis. Is that okay? Dyad simply refer to this thing we call chromosome. Refer to how many chromosomes? 2, right? Those 2 are called what? Sister chromatids. They are called what? Yes. I'm going to explain what chromosome is in a jiffy when we talk about. Just get these 2 are sister chromatids. So when they now come together again, this guy will now come together with another chromosome. They will now come together. They will now form 4 legs. They will be attached together here. Do you understand? They will form what is called the synaptonemal complex. Well, just leave it at that. Synaptonemal complex. I explained that in that other video. So there's a video on meiosis. So yeah, you're going to watch it. But I just want you to know that now there's a point where they are attached together. This point of attachment here is called what? Centromere. Do you remember? Centromere. Okay. But the point is this now. The point is this. These guys come together in what? In zygote. Zygote. Now in pachytene, something very unique takes place. Now, pachytene or pachinema, is that okay? That's the noun. This is the adjective too. In pachytene stage, something very unique takes place, and that is what is called recombination. It's called what? So this is where traits and characteristics get exchanged. Now, we define genetics as the study of heredity and variation. So heredity is what? Transfer of what? Characters from parent to offspring. Another name for offspring is progeny. You know that, eh? Offsprings are also called what? progeny. So when you transfer traits from offspring, parent to progeny, that's heredity. But variation is now this. So you look like your father, right? But you don't look exactly like your father, right? Your brother will look like you too, and both of you like your father, but you will not look exactly the same, right? So those differences between you is what we call what? Variation. Is what we call what? Variation. That's what we're studying here. Now, why those variation? Why that variation actually exists is because of what we call recombination that occurs here. Is that okay? Recombination, or another term for it is crossing over. Or what? Crossing over. Crossing over. How does it occur? Simply like this. So this one is from the father, this one is from the mother. Is that okay? Now, as they have synapsed together, the synapse here, is that okay? The process is called synapses. The process is called what? Synapses. So they have synapsed here. Now, when they synapsed, the synapse at this portion here, what now happens is this. Recombination is the process by which those portions that are synapsed together, they now exchange their genes. So the genes will now mix. So the gene from, let's say, this portion touched each other. This portion touched each other. So from this side, some of the genes on this side will come to this side. Some of the genes on this side will come to this side. Is that okay? So they will mix up that way. So and the process is called what? Crossing over. Is that okay? And the points on the chromosome that crossing over takes place is called chiasma. It's called what? Chiasma. Then if it's plural, it's called chiasmata. Is that okay? Chiasma. So we understand. So it is this. So the weights. So the genes that will mix up, they assort themselves independently. When we get to Mendel's law, you are going to understand that. So the genes that come together, they assort themselves. They mix up anyhow. No, not anyhow, in a specific way that is different from the way the next set of genes will mix up. Is that okay? That's why the genes that mixed up to form you are different from the genes that mix up to form your brother, albeit they are from the same parents. Do you understand? So if you are tall, your height might not be exactly the same as your brother. Is that okay? So because of there are other conditions, that lead to, that control those characteristics. But the point is that is exactly why you don't look exactly like your parents or like your other siblings. Do you understand now? Because of what? Recombination. And when they now separate at so prophase, the next thing is what? Metaphase, right? What happens at metaphase? I told you to remember P for prepare, M for middle. Is that okay? Then P mat, right? A for away, God bless you. T for tear. Good. So in metaphase, they will now arrange themselves at the equator of the spindle fiber, right? They will arrange themselves at the equator of the spindle fiber. Like this, like this, like this, like this. But this is tetrads that arrange themselves. Tetrads that arrange themselves here. So by the time, so this is at metaphase 1, basically, metaphase 1. So they will now, at anaphase, what will they now do? They will now separate. So the difference between mitosis again and meiosis is that in this anaphase 1, the separation takes place between non-sister chromatids. When they came together, first, for them to come together, they have to be chromosomes that look alike. That is, they contain the same kind of gene. Is that okay? So for example, if on this chromosome we have gene for your color, the color of your skin, the gene for your height, the gene for the color, the shape of your nose, the gene for the size of your head, the gene for the size of your eyes, the gene for the size of your ear. God bless you. You understand? So these genes, if this chromosome contains that set of genes, this one too, on the mother side, so this one is from the father, this one from the mother, they contain the same set, so they look alike. That's why they came together. Is that okay? So, but by the time they are separating, they will separate as dyads. Number one, they separate as what? And they separate as non-sister chromosomes. They separate as what? Non-sister chromosomes. In mitosis, when they are separating in anaphase, they separate as monads. You notice what gets to this thing. So when they are separating, what you see is one leg, right? one arm of the distance. But in this case, when they separate at anaphase, you see something like this. The distance always leads, right? The guy always leads. The chromosome always leads with the centromere. So what do you see? Something like X that is being drawn towards, right? So this is a dyad. Does that make sense? So on this end of the pole is being pulled towards here. Then on the other end of the pole, this guy is also being pulled towards that side. Is that okay? Anaphase. Then by the time it now gets to the extreme, the two poles, what happens? Then there's a splitting. Is that okay? So my point is on this recombination. So you see, so the problems that occur with genetics, with different people in their genetic makeup, some of them, many of them are linked with this, and we're going to get to that when we get to mutation. So let's go now. Genetics. Genetics is defined, right? Genetics is defined as the study of heredity and variation. Genetics is defined as the study of heredity and variation. Yeah, you also said we're going to do nutrition. Genetics is defined as the study of what? Heredity, not hereditary. Heredity and what? Variation. Right, heredity refers to how traits are transferred. Is it transferred or transmitted or passed down? Do you understand? How traits are transferred from parents to what? To progeny, from P to P, from parents to progeny or offsprings. Is that okay? So that's what heredity studies. How traits are passed down from parents to their progeny. While variation studies what? The differences, the sum total of all the differences between between offsprings from the same parents, or an offspring and the parent. An offspring and the parents. So what that is just saying is that why you don't look exactly like your mother and exactly like your father, then of course, so that is the variation. That is the variation. All right. And someone who studies genetics is called what? A genetic. Is he a geneticist or a geneticist? I've heard someone say geneticist before, but I don't know if that is correct, but I think, well, this is correct. Geneticist. Is that okay? All right. So now let's go. Terms used in genetics. Let's start from the basic terms so that you understand everything. We're going to be treating a lot of terms so that you understand it well. But let's start with the simplest one, which is the DNA. Let's start with the DNA. You understand now? So the DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. Deoxyribonucleic acid. All right, and it contains the code for life. Contains what?
The code for life.
For life, yes. Exactly. It has a double helical structure like this. Do you remember the structure? You guys see it in movies at least. This structure was proposed by Watson Crick. Can't remember, in 19 something, maybe 1940s, or yeah, around 1940s. because quite. So this is the Watson-Crick model of the guy. Now you see this ladder-like structure here. So it's a ladder-like structure. Is that okay? We say it has a double helix. Now this ladder-like structure simply contains codes that we call nitrogenous bases. We call them what? Nitrogenous bases. And there are 4 nitrogenous bases on the. I'm trying to take you through nuclear DNA now. Is that okay? If you don't know this before, raise your hand. What did you know it? Okay, what are the nitrogenous bases? Adenine, thymine, cytosine, guanine, and guanine. Is that okay? All right. Now, how does the DNA differ from the RNA?
I know something like
The C is what? No. Okay, let's go. Now, in the RNA, what we have is adenine, cytosine, guanine. The thymine is substituted by uracil. Is that okay? Uracil. Do you understand? So how do these ladder-like structures, how do they form? These are the form. They base pair. They do what? By base pairing. So right, base pairing. So that's just like the linkage between what? Nitrogenous bases. The linkage between what? Nitrogenous bases. And they are split into 2, these nitrogenous bases. Is that okay? We have adenine and guanine are called purines, right? Do you remember? Adenine and guanine are called what? I can't remember if it's purines. It's purines, right? Purines. I don't want to misplace them for each other. Then the remaining 3, thymine, guanine, and cytosine, they are called what? Pyrimidines. God bless you. Is that okay? They are called pyrimidines. You are going to understand it as far as you are following me. Just follow me. Now let's go to the base pairing. Now these are the base pair. So in the DNA, this is the RNA. Now in the DNA, we have adenine always base pairs thymine. Is that okay? Or thymine. Is this thymine or thymine? Thymine. Yeah, because it's THY. So then guanine always base pairs cytosine. Is that okay? So now write. Leave like 2 lines. Go and write gene. Let's quickly define gene so that you understand now something now. Gene is a segment of the DNA. A gene is a segment of the DNA that codes for a particular characteristic. A gene is a segment on the board of the DNA that codes for a particular what? X-tick. Just write X-tick. Is that okay? Okay. Now, it is formed when nitrogenous bases are taken in triplicates, in triples or triplicates. Anyhow you put that. Do you know what triplicates mean? 3, yeah? Good. So if I have this sequence, look here now. If I have this sequence on my guy, on my DNA, adenine, guanine, thymine, guanine, adenine. Now what will it base pair? What will be base pair here? Remember at GC. Remember what? So what will base pair this guy now? C. What will base pair this guy?
C.
What will base pair this guy? What will base pair this guy? Good. Good.
C.
Good. So now that gene is formed in triplicates. So in triplicates called codons. Called what? So it means that a gene here is going to be what? 1, 2, 3. Is that okay? It's called what? A codon. There's also something called anticodon. I will not explain that here. Just don't worry. So that's codon. Is that okay? So a gene is formed in what? In codons like this. Do we get it? All right. No. Gene, that one. I know that. Does that mean I should not play? Now, gene, I forgot what I wanted to say. Okay, so we now know what a gene is, right? It's a segment of the DNA that codes for a particular trait. All right. So complete this sequence for me. Okay, yeah, let's quickly learn about RNA and DNA. RNA undergoes a process. There's something called the central dogma of molecular biology. I'm just trying to go back and forth in between, because after genetics we're supposed to learn about chromosome itself and all of these things. So let's just do everything all together in one shot. Are we good? All right. So there's something called the central dogma of what? Of molecular biology. Central dogma of molecular biology, but we're not doing molecular biology. Is that okay? The central dogma of molecular biology says that from DNA to RNA, from RNA to proteins, that's the sequence of life. Do you understand me? That is what? Sequence of life. So basically what DNA codes for at the end of the day is what? Proteins. Are we good? Okay, but let's quickly go over it. Now, when DNA makes copy of itself, it's called replication. Told you, no? When DNA makes copy of itself, it's called what? But when DNA turns into RNA, it's called transcription. It's called what? So it means that if DNA is being copied to DNA, what is this process called? When DNA is being copied to DNA, replication, because I didn't say making copy of itself. Do you understand? So when DNA is being copied to DNA, so what happens is it's very simple. When this thing wants to copy here, it will unwind. It will turn to a tape by a tape again. Do you understand? To turn to a tape, it will unwind. That helical structure will unwind and become a tape, and then it will now tear in the middle, kind of. There's something that causes it to happen like that. Then it will now be adding another thing to it. So when it tears into two, there will be two parts that want to be copied now, right? So there's something called DNA polymerase. DNA polymerase will now start adding DNA what? Is an enzyme. You think it's hard? You think it's anything? I've told you, always ask me the origin of things. Poly, what does poly? Plenty, right? Polymer, what does polymer mean when we did chemistry? Polymerization. That is addition of several smaller units called monomers, right? Polymerase, when you hear A's, it means what? A proteinous enzyme, isn't it? So it means that it's a proteinous enzyme that is going to add, do you understand? Add plenty units to it. So DNA polymerase, that's the name. That's the name of the enzyme that helps in the copying of DNA. Do you understand now? So when DNA is going to DNA, we call it replication. But when DNA is now going to RNA, what do we call it? Transcription. Is it clear now? So when DNA wants to copy, so if this was in the RNA, if this was in the DNA, if it was in the DNA, A will pair with T, are you with me? G will pair with C. Is that clear? Then, but if it was in the RNA, if RNA wants to copy that one branch of that DNA that tore into two, it's not that it tore into two, yeah? So if DNA wants to copy, RNA wants to copy it, it will copy it as this. A will pair with U because there's no T, God bless you. Then G will still pair with what? C. Does that make sense? Okay, finish this sequence for me now. If I have a sequence that looks like this, complete it for me with another arm. So this is, and tell me where this is happening, whether it's DNA or RNA. Come on, who can come and finish it? Or should we do it together? Huh? Okay. So what will base pair this guy? Let's create another line here. What will base pair this guy? See, what will base pair this guy? Here, 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 G. Do you get it now? So where is this happening? Is this DNA to DNA or DNA to RNA? DNA to DNA. Do you understand? Is it hard? No. Okay. So, but if this were not there, if it were U, now it can't be U, it has to be A. So it will now be copying to what? To U. So this will be what? DNA to what? To RNA. All right. And there are different types of RNA, right? What are yes?
To know DNA as in transition to
RNA.
there must be 2
No, not 2A. As in A will base pair U instead of T.
How can we?
No. When you see the base pairs, you are going to see, once you see U, you spot U anywhere, then that is RNA. Once you spot that U are seal. Is that okay? They have their shapes, so and I don't want to bore you with that. So let's just leave it. Okay, now. So we understand how base pairing. And I said there are different types of RNA, right? What are different types of RNA? We have the rRNA, yeah, tRNA, and what? mRNA. Is that okay? Are you with me? So the tRNA, can't remember now. The mRNA is the one that copies from the, which one copies? Oh, I think it's the mRNA copies from the DNA. DNA, right? The mRNA copies directly from the DNA. Is that okay? It will now carry the message. God bless you. That's why it's called messenger RNA. What's this one? Transfer RNA. And this one?
Receiver.
There's only receiver. Is sender. Ribosomal RNA. Ribosomal. So I say ribosomal RNA. Transfer RNA. Messenger RNA. Is that okay? So who copies directly? Which RNA copies directly from DNA? Messenger RNA. Is that okay? The messenger RNA will now go to the, where is ribosome being synthesized? In the cell. Nucleolus. Is that okay? Should I quickly take you through the cell? No, after this video. So nucleolus is where ribosome is being synthesized. And ribosome is where protein is being synthesized, right? So what will happen is this. This messenger RNA will now take it to, it will take it to the ribosome. For the ribosome to, in the ribosome we now have the tRNA. The tRNA will now copy from the mRNA. Is that okay? Will now copy from the mRNA to direct the ribosome on which type of protein that it should create. Do you understand now? Is it clear? So who directs the activity of the RNA, of the ribosome rather? Transfer. Is that okay? So, and how does it direct it? By copying the mRNA. Is it clear? Alright, so, and that's the sequence of protein production there. So it's DNA that will now, so the type of RNA that is produced will now inform what kind of protein will be produced. Are we clear? Because amino acid sequence will also be copied from this transfer RNA. Are we clear? Are we clear on that? So that is all about DNA. Not that's all, though. That's just like the basic thing that you should know as a good student. Is that okay? As a good, okay, you know we have not started at all, or have we started? No. Okay, now let's take what is called, allele. No. So we've talked about DNA, right? So let's talk about when DNA begins to coil up. There's something called chromatin. Wait, chromatin, chromatid. Suppose I'm following you. Chromosome. How many chromatin, chromatid, chromosome. Is that okay? Then we now have okay, we'll now talk about locus. Okay. All right, so right. The chromatin is the unwounded structure, wounded. Is it unwounded or unwound? Unwounded, right? Unwound structure. This is the unwound form. This is the unwound form of the chromosome. Chromatin is the unwound form of the chromosome. That this chromosome is actually a condensed structure. So when it unwinds and opens up its chain, it becomes what? Chromatin. Then chromatid is a chromosome that has been, so you see chromatid is a chromosome now, right? A chromosome that is separated from its sister chromosome. Does that make sense? God bless you. That one, right? So this is a chromosome, right? This is a chromosome. If I remove one, I form what? A chromatid. Those two are called sister chromatids. These two, they are called what?
Sister chromatids.
Chromatids. Are we good? So then the chromosome is the condensed structure of the DNA. Is that okay? Right. Chromosome is the condensed structure of the DNA, wound around or wound up by histone proteins or proteins called histones. It's the condensed structure of the DNA wound up by the what? Histone proteins. Wound up by histone proteins. Yes? Histones? Histones are a special type of protein that help DNA to coil around. The DNA is a very long structure. Is that okay? Can contain millions of these things. That ATGC thing can contain millions. So how does that thing? What's the size of your cell? Your cell is about how many microns? 3 microns or 10 microns? 3 to 10 microns, right? You know how small that is? That's like 3 times 10 to the power minus 6. That's how small the cell is. So how does that small thing fit? And this is the cell. The cell still has other organelles. Then the nucleus is just somewhere inside it. You understand? And inside that nucleus is where DNA is. So how does that thing? So how does that thing fit there? It has to coil, thousands of times. You know what I'm saying? Is it clear? She likes you. Millions of times. All right. So now, right. Locus. The locus is the particular segment. What's that? Is it now? Look at it. Yeah, mass. Have we done it? What is in locus? Is it not just? So draw this. Let's draw a segment of the chromosome where a gene is found or located. So look here. If I say I find a gene here, if there is a gene here, if I say gene A is here, this will be the locus of gene A. Does that make sense? Think of it as location. Does that make sense? This particular portion of this chromosome, draw it to the particular chromosome. The particular portion of this chromosome where a gene is found. Locus. He went back again. Then this point where the sister chromatids are attached is called what? I've said like 1 million times. Centromere. It's called centromeric region. It's called the what? Okay. Somebody say okay.
Okay.
You like this guy. That should look like me. If you're not careful, I'll win this class now. You are annoying me. This one looks like me, you're laughing. Are you laughing? Okay, it's funny, right? It actually looks like me. Oh. Like that's how we see it. Oh. Okay, are we there? Okay, now let's go to what is called, so we now understand all of these things about the chromosome, the gene, and all of that. Now, give me what is called a trait. A trait slash character, that's another name for trait. A trait is also called what? Or characteristic. A trait is an inheritable feature. A trait is what? Okay, that is passed down from parent to offspring, progeny. Thank you, P2P. You had P2P, right? So I like it. That is passed down from what? From parent to progeny. Okay. Mommy, how are you? Yes, ma'am. We've not talked to. From parent to progeny. That's inheritable trait. E.g. height. E.g. what? Eye color. God bless you. Shape of nose. There are like how many types of nose? I think like 4 or 5. 6. I used to know straight nose, Roman nose. Okay, you see Roman nose before? It's Roman. Yeah, colorful nose is quite Roman. Our nose is like mine. It's like my own. Your own is slightly Roman too. Slightly Roman. Anyways, shape of nose. Then the Caucasians, you know Caucasians usually have flat nose. Is it Caucasians? Yeah, Caucasians. Chinese, those guys, are they Caucasians? They are Caucasians. We are Negroids, right? And blacks are called Negroids. In race, Nigger. Do you understand now? Negroids. Anyway, just leave that. This race, types of race, we have Caucasian, Australoids, Negroids, Australian, American guys. That's you get it now. You know those guys that have long nose, long and big nose. It's very British. And I was living there too. My nose is very long. Then I came to Nigeria. I decided now with this. Koreans, right? Everything about them is small. Honestly, which ones are smaller? Is it Koreans are smaller? Chinese are smaller? No, I think Koreans are smaller. Koreans are smaller. We said trait is what? Inheritable what? Characteristics. Is that okay? Now, so I will mention some examples, right? Okay. Now let's talk about alleles or allelomorph. That's the full word. Allelomorphs. So what does morph mean in class? Hey! You didn't hear on Sunday. It's 1 year now, they change. It means form or shape. Morph means what? I remember treating this when we did amorphous and crystalline here. Amorphous, no form. Do you remember? Morphology, study of shape. Do you understand now? Study of form, structure, shape. Metamorphose, that is the shape will change. Do you understand now? Do you get it now? All those words, the only thing is simplest in the whole wide world. Do you understand now? All right. Now, allelomorphs. Now, what are alleles? These are alternative forms of a trait. These are what? Okay. Let's start with, so you understand clearly. Let's start with e.g. The G for tallness is represented by T, capital T. Put that down. E.g. the G for tallness is capital T, and for shortness, small t. Both of them they are both coding for what? What are they coding for? Height of the organisms, right? Or they are coding for alternative forms of the height. Do you understand now? So we say alternative form of the trait. Which trait are they both coding for? Height. But they are alternative forms of the height. Does that make sense now? Okay. For example, we have albino. Okay, let's just, so you understand what that means, right? So they are alternative forms. So we call it allele. Yes, they are still alternative forms of the same trait. Is that okay? Albeit, when we use X and Y, we always talk about them using chromosome. We always mean chromosome. Is that okay? So XX, XY, those are chromosome. Female and male. That's why we are the one that determines sex now. We are the one, it's not them. Yes, sir. All you need to do there is get the meaning of the words. There's no chromosome cell. What's chromosome cell? Those one that will be wrong. There's not like chromosome cell. You understand? When we get to questions, we are going to talk about those things. Okay, so we said that is allele, right? Now let's talk about dominant allele. Dominant allele and dominant allele. This is an allele that expresses itself, that always expresses itself. This is an allele that always expresses itself even in the presence of another allele. That is expresses itself in the presence of what? That always expresses itself even in the presence of what? Another allele. Jomi. She's my daughter. That's the way she looks like me. That's why I named that Jomi. Yes, no? Yes. Especially in the face. Just look at the face. How do you know? How do you know? He said she made it. Yeah, wicked. And then dominant allele is what? An allele that expresses itself in the presence of another allele. The recessive allele is one that is overriding. Okay, so this one overrides. This one does what? Why this one is overriding? This is overriding. What is it? Okay, so I said that what? Recessive gene is one that is what? Always overriding or masked by the presence of a dominant allele. It's always what? Overriding or what? Or masked by a dominant allele. It's always overriding or masked by a dominant allele. Okay. So, so mask, it means it covers it up. So the dominant allele, okay, the recessive allele only expresses itself when it is homozygous. Right? The recessive allele only expresses itself when it's what? Homozygous. And you understand what that means now. Homozygous means it has the same pair. Yes, it comes in pair. So this is the only time it can express itself. But whenever you have a dominant allele plus a recessive allele, the dominant always expresses itself. Is that okay? Now, right, homozygous and heterozygous. Homozygous is two similar allele in a gene. Two similar alleles in a gene, and it is called, it gives rise to what is called a pure breed. It's called what? Pure breed. Pure breed. So two types of pure breed for tallness, what? TT or what? Does this make sense? Homozygous, the same gene, alright. And heterozygous is what? Two different types of allele in one gene. Two different types of allele in one gene. The dominant is always expressed phenotypically. So you understand phenotype and genotype now, just calm down. So when we say phenotypically, it means, so for example, okay, right quickly, please, be fast, double faster. The phenotype, I said, this is what different allele, I'm writing rubbish, in a gene. The dominant allele always expresses itself. The dominant allele always expresses itself. Do you understand? Over the recessive. Phenotypically. Put phenotypically, that is the one that we'll find in the phenotype. Phenotypically. So what that means is this pheno is from the word, yes, sir. From the word, there's a word called phenane. Phenane, is that okay? Phenane means obvious or seen, is that okay? Something that can be seen, that is expressed, that is shown, do you understand? Alright. Or that is outside, kind of, do you understand? Something that is outside is seen, right? Okay, so that's not the phenane. So it means, so if you are tall now, you are either homozygous tall or what? Heterozygous tall. Does that make sense? Once there is one capital T there, the person will be what? Tall. And whenever you are writing it, you should always write the capital first, the dominant allele first. Is it clear? Okay, so let's go. So that's the heterozygous. Now let's write. Phenotype is the sum total of all the observable features. Phenotype is the sum total. How many terms have we taken now? Twelve. How many terms with you? You know, it's just terms, just different terms. This is a sum total of all observable features or physical features. Sum total of all observable features or physical features of an organism. Sum total of all observable features or physical features expressed by an organism. Domain. Then genotype, sum total of all types of gene. What's cleaner? Eraser. She needs eraser. Sum total of all types of what? Gene. Think of it like this. See, genotype, type of gene. Yes, sir. Look at this now. Look at this now. Look at this. Like, what is the phenotype that we see here? For tall, we are still talking about height now. The phenotypes you see here are, what will be the height of this guy? Will it be tall or short?
Tall.
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 Tall. Tall. Good. So how many talls do we have? 4 talls. That's the phenotype. What will this one be? Short. 1 short. So they are saying you have to find phenotypic ratio, not 4 ratio. Does that make sense? Does that make sense? This unlikely weight in your